Good morning. My name is Jeff Smith, and I'm the CEO of Robarts Clinical Trials. I'm so pleased that more than 900 of you have taken time out of your schedules to join us this morning for our inaugural live stream event. This is the first of a series of webinars we'll be hosting that will feature experts in the field of clinical research. Today, I'm joined by Dr. Bill Sanborn, Dr. Brian Fagan, and Duncan Hall, individuals whose vision and tireless efforts have moved their respective fields and our organization forward. Before we move to today's speakers, I want to share with you some exciting news about our organization. Over the past 30 years, Robarts Clinical Trials has grown from a unit within the Robarts Research Institute at Western University to a global organization with more than 300 employees. With a vision to transform human health through academic and private partnerships, the organization has reinvested its profits into research that aligns with a purpose shared by our partners to accelerate drug discovery. Our commitment to research and the people responsible for our success has never wavered. That focus has allowed us to expand our global footprint, deepen our operational expertise, and further strengthen our scientific networks. A little more than 2 years ago, with support from Western University, we transitioned ownership of the organization to an independent health trust. The vision of our board, our leadership team, and our former CEO and current scientific director, Dr. Brian Fagan, was to establish a structure that was loyal to our mission to transform human health and our guiding principle to use profits from our operations to build enduring businesses that prioritize research and the best interests of our employees. Operating within a trust allows our business to be enduring. We have a 100-year perspective, and to measure our success not by shareholder returns or the value created by an exit, but rather by how well we fulfill our guiding principles and purpose. In line with our continued evolution, we made the decision to rebrand the company. Today, I'm honored to introduce you to our new brand, Alimentiv. Alimentiv, derived from alimentary, connects with our vision to be a leading GI CRO that accelerates drug discovery. Its meaning, to nourish and provide sustenance, represents our values of building enduring businesses that prioritize research and people. Our symbol, an olive tree, is a connection with our deep roots. The olive tree endures because of seeds planted and harvested more than 2,000 years ago. Our organization is no different. Because of the hard work of the people of yesterday, and from the commitment and effort of our people today and in the future, we have an opportunity to grow and pursue our purpose for 100 years or more. Under the Alimentiv Health Trust, we will operate a group of healthcare companies, the first of which is our CRO, Alimentiv Inc., formerly Robarts Clinical Trials. Before we move to our invited speakers, I'd like to take a brief moment to introduce Dr. Brian Fagan, who in 1997 became the director of Robarts Clinical Trials at the Robarts Research Institute, and for more than 20 years has provided scientific and operational leadership to the organization. Brian will lead our panel discussion today. As the former CEO of Robarts Clinical Trials and now Senior Scientific Director of Alimentiv, Brian has had a profound impact on drug development and IBD. I have the privilege of being part of this event today, along with all of you, because of the effort and resolve of individuals like Dr. Fagan and others like Peggy Vandervoort, one of our trustees and past operations lead, and Cindy Wong, our former general manager and statistician. For more than a decade, these individuals and a small team of 10 to 20 committed team members, some who remain with us today, built the foundation of our organization. They established a culture which promoted a passion for innovating clinical trial methodology and outcome measures, were relentless in their work ethic, and persevered through tough times, inspired by a grand vision to transform human health through private and academic partnership. These characteristics are woven into the fabric of our organization. They inform our purpose, and they are lived out every day by the 300+ employees who now work alongside our sponsors to bring new treatments to patients with unmet medical needs. I'm privileged to have the opportunity to serve as the CEO of Alimentiv, and I'm grateful for the commitment of Dr. Fagan and the early pioneers of our organization. Our keynote presentations will be followed by a panel discussion. All lines are muted, however, so you're invited to submit questions using the chat box on your screen. We will select questions for the panel, and any questions that we have which we're not able to address due to the time restrictions will be answered after the session by email. Thank you all for joining us today, and I would encourage you to visit our new website, alimentiv.com, to learn more. We have a full agenda, including presentations from Duncan Hall, founder and CEO of Triumph Research Intelligence, TRI, and Dr. William Sanborn, one of the world's foremost experts in the management of ulcerative colitis and Crohn's disease. I first met Dr. Sanborn in 2011 with the opening of our San Diego office, and have had the privilege to work with Bill in his capacity as a senior medical advisor to our organization for almost a decade now. Dr. Sanborn directs the Inflammatory Bowel Disease Center at UC San Diego Health, where he is chief of the Division of Gastroenterology, vice chair of clinical operations for the Department of Medicine, and a member of the Clinical Practice Oversight Board for UC San Diego Health. Bill is a professor in the Department of Medicine at UC San Diego School of Medicine and conducts clinical trials which have been instrumental in developing modern treatments for IBD. Bill joins us today to speak about the emerging landscape of GI drug development. Dr. Sanborn will address challenges such as high competition, payer exhaustion, limited methodological innovation, and the wealth of new agents, and offer his insights into the role innovation and partnership can play in overcoming these challenges.
Thanks very much, Jeff, and good morning, everyone. And it's my pleasure to review our opportunities for clinical trials going forward. All right, so what are the problems? As Jeff said, it's a highly competitive landscape. There's payer exhaustion. We really haven't seen much evolution in the methodology of clinical trials for decades, and there's so many new agents. So let's go through these in a little bit of detail. So we'll start with recruitment rates. Here you can see the recruitment rates over time for a decade or more for ulcerative colitis and Crohn's disease. And what you can see is that you've gone from 3, 4, 5 patients per site down to about 0.13 patients per site per month and less than 0.1 patients per site per month for ulcerative colitis and Crohn's disease, respectively. So you can expect in the current clinical trial to get about 1 patient to 1/2 a patient per site per year. And it's easy to see that this is sort of not a sustainable trend. And here's another way of looking at it. The blue represents the number of opened clinical trials in 2008, and then the blue and the white together represent the number of clinical trials in 2018. And you can see the just dramatic growth in the number of clinical trials that are going on by geographic region over a 10-year gap. What this doesn't capture is the dramatic increase in the size of the clinical trials as well. So it used to be that a big development program might have 500-600 patients. Now it's not uncommon to be 1,500 or even 2,000 patients or more. And then we have the payer issue. So this was an interesting paper that was published earlier this year. It came out of work done by the Crohn's and Colitis Foundation. They looked at a commercial insurance database that covers about 40,000 or 40% of covered lives in the U.S. And what you can see is the IBD patients in the dark gray and control healthcare expenditures in the light gray. And you can see these huge differences in the total direct cost per patient per year, which can be in the sort of $25,000 range, the out-of-pocket cost for patients, and the total lost wages. And any way you want to measure it from the payer perspective or from the patient perspective, having IBD is really expensive. And then you can see the cost over time, and there's always been a gap, but the difference between controls and the disease was the delta was pretty steady over the years. And then about 2014 or so, as we moved to multiple biologic agents, you can see the gap growing, and we don't know where the gap between patients and controls for cost is going to plateau. And then if you get into the details, here's what's driving it. It's the cost of biologic medications, which are just going up as a fraction of the total healthcare expenditures and driving that rising delta that we saw in the previous slide. And then, we haven't done much different in how we conduct the clinical trials. So, we think back to the first pivotal study. It was a phase 2 study that Steph Targan led, comparing infliximab to placebo in Crohn's disease. It had 25 patients an arm, a total of 100 patients in the trial, and it was 4 weeks long. And we've adapted that. It can be bigger and it can go for longer, for 8 to 12 weeks. But that so-called short-term induction trial, which we often refer to as the Targan design, sticks with us today. And then following out of that with infliximab, we had open label induction and re-randomized maintenance. And that heavily sticks with us today as well. In the lower right corner, you can have induction and take all the patients and re-randomize them to maintenance. That was first done in IBD by Robarts Clinical Trials when Brian was studying methotrexate in Crohn's disease. And then we have the so-called treat straight through design, which looks more like a rheumatology trial where you just treat patients, not artificially breaking them into induction and maintenance out through 6 months or more commonly a year. And all the drugs we have in the market have done some variation of these designs, not a lot of methodologic change. And then there are some opportunities. We've basically focused on moderate to severe ulcerative colitis and Crohn's disease. There was one attempt at looking at postoperative Crohn's disease recurrence. The trial failed really because of outcome measure issues by regulatory authorities, but this is an opportunity going forward. Mild to moderate Crohn's disease, the last drug approved was in the late 2000s in Europe, or late 1990s in Europe and early 2000s in Crohn's disease, Entocort. And in the meantime, the FDA has imposed endoscopic endpoint. And with that in mind, we don't really know how to study mild to moderate Crohn's, but this is both a challenge and an opportunity. We have fibrostenotic or stricturing Crohn's disease, which work is going on now. That's a new frontier. Isolated distal ulcerative colitis or proctitis, very little work in that arena. That's an opportunity. Pouchitis after colectomy for ulcerative colitis with creation of an ileoanal pouch, nothing approved for that. There's some methodologic challenges for outcome measures, but it's an opportunity. And then there's the liver inflammation or primary sclerosing cholangitis that occurs in about 10% of patients with ulcerative colitis. Again, nothing approved for that concomitant disease that we see often in IBD. And then there's just a lot of new agents. For those who are on the line today, if I forgot your molecule, I'm sorry. There was just only so much room in the slide. We have anti-integrin therapy. Anti-beta-7 is coming along and reads out this summer. We've got a number of small molecules and peptides that are orally administered in that space, and we're waiting to see more definitive data on that. We have a pan-JAK systemic inhibitor, tofacitinib, approved for ulcerative colitis, but you can see this long list of JAK1 and JAK3 selective drugs, TYK2 drugs, and topical pan-JAK inhibitors in the JAK inhibitor space. Lots of activity there. We saw a press release that the phase 3 trial of ozanimod in ulcerative colitis reached all of its primary and secondary endpoints for induction and maintenance. So that, I think, means that we will have an S1P1 modulator in about a year for treatment of ulcerative colitis. We have more drugs in the pipeline. There's LAG-3 study in ulcerative colitis. There's 4 drugs, 3 or 4, looking at anti-interleukin 23 or anti-P19 therapy. There's microbiome therapies, HIF-1 alpha stabilizers, anti-TL1A therapy, topically administered cyclosporin, microRNA treatment administered orally, TL1-9 or TLR9 antisense and LANCE2. And again, I know there's other things that would also be in this list. So many drugs to come. So what can we do about this? I'm sort of ready to go have a drink after all of that, but that's not the way forward. We really need to innovate out of these challenges. So what could we do? And I thought that there's a number of possibilities that fit broadly into the areas of innovation and outcome measures and clinical trial design, strategies for achieving greater efficacy, and then sort of thinking about GI diseases, alimentive, which in part, as Jeff said, has to do with alimentary tract or gastroenterology, there are other diseases in the GI and liver disease space that may be of interest as well, where the lessons that we've learned and are learning in IBD clinical trials could leverage into some other GI therapeutic areas. So we'll start with innovation in disease activity assessment and outcome measures. And, this is, I liken this to writing the code for whatever your favorite Microsoft or Google or Facebook application is. Most of the work goes into making the architecture for the platform, and then the platform can sort of take off. And making the architecture is a bit boring, but it's really essential. The innovation in this arena at Robarts went back to the 2000s when we conducted a placebo-controlled induction trial of a new mesalamine formulation in a number of far-flung jurisdictions. And in those days, the internet wouldn't support compressing multi-gigabyte videos and transmitting them over the internet. So we had the local site's video with our computers, the procedures, put the videos onto a memory stick and ship it to Robarts and have it centrally read after the fact. So the patients were randomized based on the local read, and we got an overread in all the patients. And here on the left, you see modest differences for clinical remission, and at week 6 on the far left was the primary endpoint, and that 9.4% or 9.4% difference had a p-value of 0.056, so it missed. And in principle, and the drug ultimately was not approved in the United States because it missed. But we have the opportunity to take all those memory sticks and have Jack McDonald reread all of the videos. And you can see that the delta improved significantly, and we had multiple zeros in the p-value with the reread. And what we found was that 31% of the patients actually didn't have active disease at baseline. And if you remove those patients, even though you cut the trial size down by about 30%, that you had highly significant results. And this told us that there were a number of biases to having local readers that if you centrally read, you could overcome and make clinical trials more efficient. And going forward from there, now basically all clinical trials have central reading for both endoscopy and both ulcerative colitis and Crohn's disease. We then moved into Crohn's disease. There were a couple of off-the-shelf instruments, the CDEIS and SCSCD, and we looked carefully at these and found that you could reliably read both instruments, but there was better agreement or better kappas between readers if you looked at the SCSCD, which is a somewhat more simple instrument, and that's come into widespread use in clinical trials now. And then we moved into histology, and there wasn't so much an off-the-shelf instrument. So we had to sort of take some existing instruments and go through, really start from scratch and develop and validate a new histologic index. In this case, the Robarts histopathologic index. Another instrument was developed in parallel in Europe, the Nancy index, and we've extensively studied the operating characteristics of these two instruments. This one has, the RHI has two utilities. One is looking at changes in mean score, which you can do in smaller numbers of patients. I'll come back to that in a minute. So we think it's responsive and well suited for early drug development. And then you can read it in a dichotomous fashion, and it's well suited for late stage development. And again, scan slide centrally read. And then MRI, there was a lot of innovation in Barcelona, led by Julian Panis and Jordi Ramala, in Barcelona, around developing MRI enterography indexes. We collaborated with the group in Barcelona and other radiology experts around the world, and refined a MRI index and showed that you could make it work in a multi-center fashion. And this is really ready prime time now in Crohn's disease trials. We have the same thing in fistulas. So we've looked at both MRI enterography for luminal Crohn's disease, and a novel instrument for perianal Crohn's disease. And then talking about early drug development, this was work led by Vip Jareth, where we were looking for a continuously expressed instrument with a wide dynamic range where you could look at changes in mean score by treatment group and see effects in phase 2 trials in smaller numbers of patients. We're working now on a similar instrument for early development in Crohn's disease. And then this is something we're quite proud of. It's took a number of years to develop. It's a new patient-reported outcome developed according to all the criteria that the FDA laid out for how you would develop PROs. And the symptoms and impacts questionnaire applies, as it turns out, to both ulcerative colitis and Crohn's disease. And here's some of the domains. I won't go through all of it, but if you look on the left, you can see there's really sort of a stool module, but instead of just asking how many stools the patient has compared to their baseline, you can see that this really gets into the details of the stool experience, the consistency, the urgency, bleeding, and all the subcomponents, and you're able to tease those things out. You can see a pain score and a nutrition component and a fatigue component. And it turns out that over 80% of the domains are homologous between ulcerative colitis and Crohn's disease. So rather than having unique scoring systems for each disease, you can have the same overall questions and you just have different scoring systems for the two diseases that capture the relative importance of stool frequency and rectal bleeding versus abdominal pain. So what about clinical trial methodology? I think how you do late-stage clinical trials is fairly well worked out, at least for moderate to severe disease, although we're moving into active comparators. We have strategy trials that we'll talk a little bit about. But trying to use the strategies that you use in late-stage clinical development for early clinical trial development drug development doesn't work very well. So we've spent a lot of time thinking about how to innovate in that arena. And here's a paper that we published a few years ago in Gastroenterology, sort of a think tank piece around this. We subsequently published another paper that's analogous for Crohn's disease. And in panel A at the top, you can see the typical thing where you have up to 100 patients and typically 250 to 300 patients in phase 2, and then many hundreds of patients, up to thousands of patients in phase 3. And in panel B, how we think that you might approach this using the patients in phase 1 and then having much smaller numbers of patients in phase 2 to prove the concept, find dose, and then move more efficiently into phase 3. And here are some of the principles that go into that. Centrally read endoscopy, which takes away the bias component. And you can measure that not only in a dichotomous fashion, comparing the proportions of patients who have differences in endoscopic improvement or endoscopic remission, but you can also measure it in a continuous fashion, looking at mean index scores. Same thing with histopathology, taking the Robarts histopathologic index as an example. You can look at the proportions of patients who have centrally read histopathology, or you could look at the mean scores by treatment group. We've come to understand, and the field in general understands this well. Robarts has made contributions to the area of pharmacokinetics and differences in drug clearance and drug exposure. This seems to be particularly true in monoclonal antibodies. And so we think a key part of early drug development that's been underappreciated, at least with monoclonal antibodies, is pharmacokinetics and adequate drug exposure, adjusting for differences between patients. And then the pharmacodynamics and target engagement. There are a number of drugs that have come to market where the target engagement in the tissue was never really adequately studied. And our view would be these days that you want to show you're getting the drug into the tissue, engaging the target, and having the expected signaling changes from engaging that target. And finally, in early drug development, focusing more on continuous scores that you can look at mean changes by treatment group rather than dichotomous outcome measures. And there's a number of innovative things. This was one thing that I'm going to give you an example in the next slide, but that's starting to come into play, is taking a Bayesian approach to early clinical trials. So you'll have some sense in the literature, from the literature, what the placebo rate for the outcome measure that you're looking at will be. And you can continuously assess for the active treatment how it's playing out against placebo using that anticipated placebo rate from the literature. And here's an example of an anti-interleukin 17 alpha antibody that was studied in Crohn's disease. This drug is approved for psoriasis, but in Crohn's disease, you can see it looks a bit flipped around. The CDAI got worse in the patients who got drug relative to placebo, and the trial was actually stopped for harm. So this was an example of getting an answer with a relatively small number of patients, just about 60 patients using Bayesian techniques. Well, you could say, yeah, but was this really the truth? But it turns out there was a second anti-interleukin 17 drug, brodalumab, that did a larger trial using conventional endpoints and found exactly the same thing. So what was found with this Bayesian trial actually was true, that it flared Crohn's disease and made things worse. But as a broader principle, you can see the utility of Bayesian approaches. And then adaptive clinical trial design. This has been done a little bit in IBD, but not nearly enough. So this is the idea that you set out with your anticipation from the literature of what you think the active treatment is going to be and what the placebo rate is going to be for the outcome measure. But then you do an interim analysis and you pre-specify some adjustments. So which on one extreme could be futility and cessation of the trial. In the middle, just continuing what you thought you were going to do. And on the other extreme, that you can see you're headed in the right direction, but you're not quite powered, and you repower the trial. And you're able to do this if you pre-specify it without an alpha penalty. And I think this has a huge amount of promise for the future in IBD trials. And then this was a brilliant paper written by a senior official at the FDA talking about different master protocols. This has been mostly done in oncology, but it's ripe for IBD, umbrella trials, basket trials, and platform trials where you're putting multiple drugs in, sharing placebo. You start out with placebo versus one drug, or drug A versus drug B. That doesn't work. You just keep rotating through in this large umbrella of clinical trials. So patients come in, and even if they fail the first therapy, you keep them on drug by just switching through other drugs in the master protocol. And just to give an example of a few applications of these principles, recently we published looking at TD1473, which is a topically administered pan-JAK inhibitor. Used all the centrally read outcome measures that we just talked about. You measured drug in tissue, you measured phospho-STAT3 signaling in tissue, and this had about 10 patients in arm, and you could see enough from this small, early, innovative trial design to move into phase 2B. This was another interesting study looking at an orally administered peptide against alpha-4 beta-7. There were some issues with the initial central read, and the organization that did the central reads switched, and when it was reread with the validated structure for central reading, you can see a nice dose-dependent response for remission, with the highest dose having a remission rate of 19% and 6% on placebo. And here we have 16 patients in arm. You could see differences in histology and a variety of other outcome measures. And this has led to this target now moving into a more definitive phase 2B study. And this was a study published recently with an S1P modulator using the mean modified Mayo score expressed continuously as the primary outcome measure. and you could see a highly significant p-value for looking at the changes in the mean modified Mayo score for drug versus placebo with dose response shown as well. We think that the Robarts histopathologic index, the UC100 index, and maybe the total Mayo score might work even better in this arena. And a lot of work is going into trying to find the most efficient ways to look at continuous measures in both ulcerative colitis and Crohn's disease for early trials. And then there's active comparator trials. You all know the SONIC trial showing that infliximab was more effective than azathioprine or monotherapy. And recently, trials showing that vedolizumab was more effective than adalimumab for ulcerative colitis. So we expect a lot more of these active comparator trials, in part because there's so many agents that are coming to market, and we need to know how to position them relative to each other. So this is an innovation in clinical trial design. And then there's strategy trials. So with cluster randomized trials, you randomize centers or entities rather than patients, and center A has one treatment design and center B has another treatment algorithm, and you compare strategies so you're not locked into one drug. Robarts has been a real innovator in this space, actually in several therapeutic areas. And perhaps the best example is this paper published in The Lancet a few years ago, the REACT-1 trial, that showed that organized, protocolized care was better than usual care for outcomes such as differences in surgery, differences in serious complication rates, differences in hospitalization, and especially differences in a composite of all those big ticket outcome measures. Then we have several ongoing strategies. REACT-2 is randomizing patients to treating to a clinical algorithm, versus treating to an endoscopy-based algorithm. All the patients have finished this trial. We're just sort of cleaning up the data. We should see the analyses soon. And the VERDICT trial, which just started, is in ulcerative colitis and is assessing whether you get the best long-term outcomes if you treat patients to endoscopic improvement versus histologic healing. What about greater efficacy? Well, there's different ways to do this: combination therapy, precision medicine, local delivery of drugs to the GI tract, and you can mix all those strategies. So combination therapy, we already talked about SONIC that showed that azathioprine versus plus infliximab was better than either drug alone. And then a number of us have been starting in clinical practice to try mixing different biologics. And on the left, you can see there's a trial of vedolizumab plus adalimumab plus methotrexate. There's a trial of gasecumab plus golimumab. So there are now some phase 2 studies going on looking at mixing biologics. And I think there will be some winners out of that going forward. And then there's precision medicine. So this is just recognizing that there are over 250 genes now that have been associated with inflammatory bowel disease. And in that is going to be a number of subgroups where a particular inflammation pathway dominates. And if you can pick out the patients and get the right drug to the right patient, you might markedly improve efficacy. This relies on large biobank and biodata existence, and then the multi-omic approach to integrate the findings to find the important pathways for particular disease presentations or phenotypes, and then drug them. And there's definitely work going on in this arena now. And then there's just some obvious things, like we know that the biology is different for ileal Crohn's disease versus colon-dominant Crohn's disease, and yet we just keep calling it Crohn's disease. My group recently put a think tank piece out. Should we think about splitting Crohn's disease into Crohn's colitis versus ileum dominant Crohn's disease, ileitis plus right colon disease, have different outcome measures, and just think of those as two different diseases. And then there's local delivery of drugs to the GI tract. We know that budesonide and mesalamine work, but people are looking at JAK inhibitors, alpha-4 beta-7, HIF-1 alpha stabilizers, cyclosporin, TLR9 antisense, delivery of antibodies to the gut, peptides to the gut. Some of these are going to win, and I think we're going to see a big expansion of local delivery, which you can imagine from a safety perspective enhances combination therapy that we talked about earlier, and still you could have precision medicine fit into this. So I think these three things that I just talked about are going to interact in different ways. And finally, what about other diseases? Robarts is active now in developing outcome measures and starting to participate in clinical trials for eosinophilic esophagitis. There's some very innovative work that you're going to see in major publications in the coming months around eosinophilic gastritis and duodenitis that turns out may be an important cause of non-ulcer dyspepsia. You have non-alcoholic steatohepatitis or NASH and celiac disease, and we're active in all of these arenas. So in summary, there are many challenges at present in the IBD landscape. The competition will increase, and we need to bring more cost-effective treatments to the market for existing indications, develop new indications. I gave you a list of those. I think that innovation and methodology, especially in early drug development, will be essential in screening compounds to make these things happen. And Robarts certainly recognizes that effective partnerships related to these methodologic advances are one strategy for increasing the likelihood of success for all the stakeholders. Thank you.
Thanks, Bill. And if you haven't already done so, if you have any questions, you want to use that chat box feature, submit your questions. We will perhaps address some during the panel, and what we don't get to, we'll certainly respond to by email. I'm pleased to introduce our second guest, Duncan Hall, CEO and founder of TRI, who's joining us from his office in England. I first met Duncan several years ago and immediately took note of his approach and thought leadership in risk-based quality management. Over the past year, we have partnered with Duncan and his organization to build an RBQM platform called Alimenta Forte. And we'll give the opportunity over now to Duncan to hear from him as he talks to us about the future proofing of clinical trials with RBQM. So welcome, Duncan.
Thanks very much, Jeff. I appreciate the introduction. It's a pleasure to be here. So today's session, we're going to be talking about future proofing of clinical trials using risk-based quality management techniques. As Jeff said, my name is Duncan Hall. I'm the CEO and founder of Triumph Research Intelligence, and I will be taking you through a pretty short, hopefully brief presentation today. We've got four main areas we want to talk about. First of all, just to explain what risk-based quality management is. Not everybody fully understands it, so I want to make sure that we get that defined and hopefully to understand why it's so important. What problems are we trying to target with RBQM? And ultimately then, how does solving those problems drive value for sponsors of clinical trials? Anti-fragility is a discussion that's been going on a lot over the last few months for obvious reasons. And I think a lot of companies running clinical trials now are really starting to think about how we protect ourselves in the future and make sure that we don't suffer some of the slowdowns and delays that we've seen in clinical trial research during the COVID-19 driven lockdown period. So how can RBQM help drive that anti-fragility and robustness into clinical trials? And then, as Jeff just said, Alimentiv have been building a really world-class, very, very comprehensive and solid RBQM capability over the course of the last year. Let's talk a little bit about that and show some of the work they've done. So first of all, what is RBQM? RBQM is really a bit of a change in mindset. It's looking at quality from end to end. So it is thinking about quality right from protocol design all the way through to study completion. It's a move away from more traditional monitoring techniques, and it's driving monitoring based on risk. So it's thinking very strategically about monitoring, and it's helping us understand where the risks are and then driving monitoring resources to those areas and moving away from the traditional monitoring methods, which tend to be more schedule driven and maybe event driven rather than data driven and risk driven. And RBQM drives us to build a quality management plan which uses all elements of what we have available from a data perspective. So it looks at the data holistically. It allows us to use statistical methods to drive trends and models, look at outliers, and ultimately to drive insights into what's going on during the course of that clinical trial, really early on and continually throughout the life cycle of the clinical trial, which allows us to take action when it's appropriate and when we can actually have a genuine influence on the rest of the study. So what are some of the problems then that we're seeing with clinical trials today? Well, the first real problem is speed. So we've seen hundreds of clinical trials being either stopped completely over the last few months or slowed down dramatically. We've seen sites really being burdened with dealing with the fallout of COVID-19, often unable to conduct any research or dramatically reduced levels of research because of the pressure they're under to treat the victims of COVID-19. And then obviously as we come out of lockdown and we're in a world of social distancing, the ability the throughput that sites are seeing is much, much lower. Now, unfortunately, while those delays and slowdowns have been occurring, the patent cliff is just getting closer and closer. So for companies that have started clinical research, there's even more pressure to pick up the pace once we come out of lockdown. And that often puts pressure on the design and conduct of clinical trials. We often see protocol designs being pushed through really quickly. We see the startup process being pushed through. And often some of the key principles that we're talking about today, quality management and risk management and risk assessment, get completely overlooked. And that can result in poor quality, which ultimately results in cost. Having poor quality, having perhaps a poorly designed protocol can mean multiple protocol amendments, which mean further costs and delays, potentially having to make adjustments halfway through a study, taking on more patients, opening more sites. All of those have significant increases in cost, which we're really trying to avoid right now. And then the error component, so systematic errors. Any clinical trial is going to have systematic and random error. Systematic error is often caused by things like behavioral issues, lack of understanding of a protocol, equipment failures, the way that data is handled, and so on. The failure to spot those errors and understand what their root causes are early is likely to cause quality issues downstream. And again, those quality issues can then cause delays and increased costs. So we've got that classic sort of cost, quality, and time triangle that anyone that's ever done any form of sort of project management training will be inherently aware of. And in today's really challenged environment, that cost, quality, and time triangle is more important to control than ever. And that's really where RBQM comes in. So what are some of the regulatory drivers then to RBQM? So we've been talking about some of the reasons that we want to take on RBQM. Many people don't necessarily realize that RBQM has been part of regulatory guidance now for many years. In Europe, it was adopted in 2017, in the US 2018. This year, we've seen most of Asia catching up with adoption. So RBQM has been in the regulatory guidance for multiple years now. And this slide simply shows some of the key statements which are driving the adoption of RBQM. Some of the key statements there. Beneath this, there's a very detailed set of processes which really describe the process in ICH E6R2. But regulatory reasons are one of the key drivers that companies are adopting. So if we go and look at the timeline then, as I said, you can see there that R2 has been in play now for a couple of years. But coming behind R2, we've got now a revision of ICH E8, which are the general principles for clinical trial conduct. That's going to be increasing the scope of some of the RBQM principles. It certainly underpins a lot of what's in ICH E6R2 by talking a lot more about critical thinking, quality risk management, and in general, driving a quality culture through your business. But then you can also see that we've got a further revision of E6. So while some companies are still running and getting implemented with E6R2, we've got a third revision coming up, and that's due to really kick off next year. But the ICH have already started to release some initial information on what's going on with that particular release. There's not a lot of information out there at the moment, but I'm going to quote literally from their website. E6R2 included a focus on a proportionate risk-based approach to the design and conduct of clinical trials. E6R3 will be designed to further advance this concept and to encourage relevant parties to utilize this approach. So both E8R1 and E6R3 are very much going to be building on the principles of quality risk management. And I think it's very clear from that, the fact that we're getting more and more drive towards this direction, that it's here to stay and something we should really be embracing. So let's talk a little bit about what RBQM is then. You can see from this, this graphic, we've got some of the key processes that ICH E6R2 actually describes. RBQM always starts with risk assessment. So at sort of 12 o'clock on the schematic there, we start with risk assessment. That drives how we're going to think about risk management. So how we're going to manage the risks that we've identified, how we're going to report and monitor those during the course of the study. That then moves us into thinking about quality. So once we've understood what the risks are and how we're going to control them, we can start to think about quality. And ultimately, one of the key quality management techniques is going to be how you monitor the clinical trial. And one of the big changes with ICH E6R2 is the movement away from routine site-based monitoring, primarily central monitoring and remote monitoring. So if you just move the slide forward, then you can see some of the red dots on the slide show the processes that we're using technology to support as well. And this is the backbone of the Alimentiv Forte platform that's being implemented at Alimentiv at the moment. So they're performing their risk assessments, they're doing their risk reviews and their risk reporting on the Opera technology. If we move to the next slide then, thinking about the monitoring approach, then you can see the blue circles on this screen. When we're thinking about monitoring, what RBQM is asking us to do is not just to assume routine on-site monitoring, as we've often done in the past, but to think about where central monitoring and remote monitoring could be used, and how those techniques will actually enable us to identify potential issues and challenges with data and quality and safety much earlier. And if you do the final build on this slide, you'll see then the final set of red dots there, some of the key techniques within central monitoring that allow us to really be looking at that data holistically and understanding where some of the challenges are. And again, the red dots here signifying that those are processes that we're able to perform using technology in order to look at the data centrally, understand where the challenges are, and then focus our monitoring resources accordingly. And that's, again, core to the Alimentiv Forte platform that is being built at the moment. So what's the value of all this then to anyone conducting a clinical trial? Well, quality, the Q in RBQM is quality. This is all about building quality in from the beginning. It's about considering the design for the study, optimizing that design early, and then putting together a plan to continually manage quality throughout the life cycle of the clinical trial. And that's what's going to drive down the costs and increase the efficiency of the study. Strategically, then, RBQM allows us to really think about what's important. It allows us to look a long way down the road and really think about what might happen, what the challenges to this trial could be, and how we can use data and processes to control and manage those. So we're much more strategic about study conduct than we used to be. Going through those processes, looking at the data centrally, using some of the techniques that we just showed on the last slide, allows us to gain insights that we've never had. So we're not going to sites now hoping to find something. We're going to sites, or we're doing remote monitoring visits with a clear view of what's probably going on. We already know what the issues are. We've already seen what the data looks like. We've got a hypothesis, and we can use our valuable monitoring resources to go and test those theories and to challenge things and raise things with sites and understand specifically what's going on. So we have an insight that we never had before. And then finally, the cost. Obviously, costs are a challenge at the moment more than ever this year. Site monitoring is an incredibly expensive component to clinical research. And if we can ease back on the site monitoring, if we can take those expenses, all those travel expenses, all the time in hotels and on airplanes, and use our monitoring budget much more effectively by looking at the data centrally and conducting activities remotely, we can actually end up doing more monitoring than ever, getting a higher quality result, but with a much lower cost base. And so those four areas really are key, the key value statements for risk-based quality management. So how does all this influence fragility and robustness then? Again, as I said, a key word in today's environment. Well, generally the simpler something is, the less fragile it is. RBQM teaches us to really focus on what's important, to simplify where possible, to remove unnecessary data collection, unnecessary processes from protocols where possible, just focus on what the core driver for that clinical trial is. It enables us to reduce site monitoring. So by doing more monitoring, but doing that monitoring centrally and remotely, we can reduce the dependence on site monitoring. And of course, one of the key reasons that a lot of studies have slowed down now is that site monitoring and obviously patient visits have been heavily compromised because of COVID-19. So if we can look at other ways to get access to patients, we can look at other ways to monitor without needing to be face-to-face on site, we can take a lot of the fragility out of clinical trials. We can use the risk assessments that we're doing to drive monitoring strategies so that we're really focusing on what's important and that we're not overlapping between functions. We haven't got clinical operations monitoring the same data that data management have already looked at. And we can start to get much more creative. We can use critical thinking to really think about better ways to conduct clinical trials, and having that solid risk assessment and views on what might go wrong and where the challenges in a study are allow us to be more critical, but also to be more creative in how we conduct clinical trials. And so finally, what does this all look like in terms of Alimentiv? If you want to go on to the next slide. The solution that Alimentiv are building now, which is an incredibly comprehensive solution. Now, Alimentiv, I should say, have been early adopters of RBQM before it was even called RBQM. Back in the days when we used to call it risk-based monitoring, it's been years since Alimentiv have been piloting this. They've used that experience now to really build out a solid solution. We've done a complete transform of their business processes. So we've looked at the impact of RBQM on all their processes and rewritten and updated their QMS. We've implemented the Opera technology, which is our company's technology to support all those processes that we talked about. We're using this as a collaboration and a reporting platform. So all functions involved in clinical trial conduct can now have access to the same data. They can see what's going on. They can report their findings, develop actions and manage those actions in the platform. But we can also share that information with the sponsors of studies as well. So we've got one platform. There's no multiple views of data anymore. One platform that allows all parties to collaborate. They're creating an incredible training capability at the moment, which actually is going to allow end users to get exposure and experience of data, looking at data and making decisions before they go on to clinical trials. And there are a lot of mid-trial support as well to make sure that they're really getting the most out of the technology and that we're making great decisions during the trial conduct. The platform enables a full set of audit trails and inspection readiness, so we can show the data at any point in time that anybody wants to look at it. We can show that we are following a quality management plan, that we've got a plan in the first place as required by R2 and that we're following that plan. And then all those things finally enable us to really take a much more of a partnerships approach. Alimentive are able to partner with their customers much earlier in the process. They're getting involved in those trial designs, in the protocol reviews. They're basing their study conduct plans and therefore their resource plans and their costs on risk assessments, on putting together a strategic monitoring plan, on really thinking about how they're going to manage quality through better monitoring techniques. And that's building much earlier relationships and giving their customers confidence that they've really got their arms around the way that this study is going to be run and to get the best possible outcome. So all of those things are components of the Alimentive Forte solution, and that's the solution that's going to be rolled out and involved on every study going forward at Alimentive. So hopefully that gives you confidence that they've put a huge amount of time and effort into this, that's going to be something that really helps drive forward. And finally, I just wanted to say thank you. I look forward to the questions later, and I really appreciate being asked to talk today. Thanks very much.
Thank you very much, Duncan. Well, we're going to move to our panel session here. I'm pleased to call upon Dr. Fagan to lead our panel discussion. Adding to my earlier introduction, Dr. Fagan is the Senior Scientific Director at Alimentive and a Professor of Medicine at the Schulich School of Medicine and Dentistry. His research efforts focus on the design and implementation of randomized controlled trials and the development, validation, and optimization of outcome measures to assess the efficacy of novel therapeutics in Crohn's and ulcerative colitis. Brian's been the principal investigator on numerous large-scale randomized clinical trials, has authored over 400 peer-reviewed articles, more than 30 book chapters, and has given nearly 1,000 invited presentations at national and international scientific meetings. Thank you.
Well, okay. Well, thanks, Jeff. We've a lot of topics covered here, and I don't know where to start, but maybe at the beginning. Jeff, for you, the name's changed, and the corporate structure's changed, the ownership. So what's the future for Alimentive, and how does it relate to the legacy of investing in research and using that as a business strategy?
Thanks, Brian. Yeah, as Brian said, the name changes today, but the structure's been in place now for 2 years, so we can continue to operate as we have for 2 years. What we can carry forward from the past is a belief that if we continue to be driven by our purpose, which is to transform human health, and those priorities, which have always been there to invest in research, and research that ties directly to the interests of our sponsors, our clients, in terms of accelerating drug discovery, that we can continue to grow a healthy business into the future. I think one of the major changes for us in shifting from ownership by the university into the trust is that it allowed us control over the financial decision making and the risk that we were willing to take on as a private CRO operating in a very dynamic space, which is constantly changing and evolving. But I think if we continue to stick to what we do well, and that is to be loyal to our niche with some expansion out into other areas of GI research, but prioritize that research in investment, then we'll be able to continue to grow the business into the future.
You indicated that there were potential for multiple companies sheltering under the trust, and Robarts, the traditional CRO, being one of those companies. What other companies does Alimentive have planned, or
Sure.
give us a glimpse of the future in that regard?
Yeah, so Alimentive is the first company under the Health Trust. We have a second company which we will be announcing before too long, probably over the summer period, which we're quite excited about it, that aligns very closely with our CRO business. We see opportunities in a few different areas, and I think over the next year to 2 years, we'll have the opportunity to launch a few new companies underneath the Health Trust.
Maybe we could switch and ask Bill. Gosh, the first part of your talk depressed the hell out of me. And I think your rhetorical question, why would anybody be in this field, was taken to heart by probably many members of the audience. Maybe you could help us a little bit, and you outlined a lot of different innovative strategies. what's the low-hanging fruit? Where do companies go wrong in early drug development, and how can they do things better? If you had to just highlight a couple points.
Well, you're asking how to get there. The real opportunity is bigger differences between drug and placebo. So unlike, say, moderate to severe plaque psoriasis, where anti-P19 therapy is very highly effective, and you have large deltas relative to placebo, in IBD, the typical drug in moderate to severe disease works in 18 to 20% of patients, and on placebo in 5% of patients. And so, the deltas are 10 to 15% at maximum, with a ceiling effect for induction of remission of not more than 20-ish%. So that means 80% of patients aren't going into remission. So that's the opportunity. And that's why IBD is still really interesting relative to some other therapeutic areas, because finding ways to adequately treat all those other patients is a large unmet medical need, and therefore a large commercial opportunity. So then everything becomes, to me, about how to grow that delta. And what inevitably happens is that drug developers are conservative people with conservative companies behind them, and it's easy to go for the tried and true. And when you do the tried and true, you can see it over and over and over with different mechanisms. You get to that same point of a 10% delta and a 20% ceiling, absolute remission ceiling effect. So I think everything has to focus on how you grow that delta. Placebo rate's going to be what it is, but how do you grow the active treatment intervention? And I think it comes back to combinations and precision medicine. So we've got all the architecture pieces kind of built: centrally read endoscopy, centrally read histology. We're working on the continuous outcome measures. But to me, the real innovation is going to be in kind of basket trials that would allow, and platform trials that would allow efficient combinations of drugs to be tested. Now, for the small companies and even large companies, it's sort of horrifying to think that you got to go to all the trouble to synthesize a molecule or build an antibody and do the tox work, and then you go into this Lord of the Flies thing and maybe you don't come out. But I think you're going into it anyway when you see all that other stuff realistically. And so a really efficient way of combining drugs or going for precision medicine early on. Brian, you've been in the field even a few years longer than me. I'd be interested in your views, but somehow we've got to get bigger deltas, and that's how I see it.
Yeah, I mean, it's either designing the trials at early phases to screen for multiple compounds to look for a killer app effect, very large therapeutic effect, or it's using continuous outcome measures efficiently in surrogates to screen for combinations and look for synergy. Precision medicine, I'm really concerned about how successful it's going to be in the field of inflammation as opposed to oncology, where of course it's been a real benchmark there. Maybe you could come back to Duncan about the theme of conservatism in pharma. You made the case, I mean, I want to buy one based on the presentation you gave, but I don't see pharma, rapidly adapting. It's gradual. So what are the barriers? What's the resistance to the processes that you've talked about?
Yeah, thanks, Brian. I guess it really comes down to change management. I mean, when you look at the solution that Alimentiv have put in place, you can see all those components. We've been looking at processes, technology, training, the way that partnerships work, business models are changing. All those things are being influenced by RBQM, and that's a lot to deal with. We're being asked to think completely differently about the way that we design and conduct clinical trials. And sadly, I think over the last 10 or 15 years, we have got into a little bit of a commoditized approach to clinical trial conduct. It's become quite routine and quite checkbox. And we're being asked to stop doing that. We're being asked to be critical, to challenge, and really think things through. And that requires a mindset that for many companies, they don't have that across the board. And so we are having to do what Alimentive are doing right now, and that's put comprehensive training in place to help people adopt that mindset, and putting the technologies in place to be able to get the views of the data that they need in order to make accurate and effective decisions going forward. And that's a lot to take on. And when you're looking at a large pharma organization that may have 3,000 or 4,000 sort of study conduct and monitoring staff, and potentially a lot of outsourced staff on top of that, there's a lot of people that you've got to change the mindset of. And that just takes time. I equate it to the movement from paper to EDC, 20 years ago. That didn't happen overnight. A lot of people are still hanging on to those pieces of paper, and it's going to take time to change that mindset once again.
Yeah, I'm interested that
Some of us still miss those pieces of paper, Duncan.
I'm glad you said it, Bill, because it was on my mind. Duncan, I'm really interested that you didn't hold out the regulatory authorities as the scapegoat. In fact, as you pointed out in your presentation, they've actually been leading this process. So it's not a situation, it sounds like, that the regulators have got their feet dug in. It's probably processes within systems within organizations.
I think there was an acknowledgement from the regulatory authorities that there was a process of change. And as always, when there's a major regulatory change, you get a fair warning. The regulatory guidance was changed a good year before the first country started to adopt it. But as always, it's human nature to leave things a little bit late. I think the regulatory authorities have acknowledged a sort of honeymoon period, where there were very few sort of inspection findings related to R2, but we are now seeing those findings coming up, and the regulatory authorities have publicly stated that honeymoon period is over, and they do expect to see risk assessments, they do expect to see quality management plans, they expect to see thoughtful, risk-based quality management. And they are pinging companies for not doing that. And we've seen examples of companies being told to go back and retrospectively put those things in place, and that can cause a challenge. So I do think the regulatory authorities are behind it now. The FDA stood up and publicly said that last year at a big event they ran in Washington, and I think that is going to drive change. The honeymoon period is over, and we're going to see the authorities really behind this now.
Okay. Jeff, you talked a little bit about the structure of Robarts and how it is somewhat unique in regard to the interactions with academia, and that academic leaders such as Bill really have been very instrumental to our success. Can you give companies some advice about how to interact with academic institutions? And
Wow.
yeah, make it.
I'm looking forward to hearing that.
50 words or less.
Wow, 50 words or less. Well, I think for me, when I joined Robarts in 2011, it was a new environment. I hadn't come from a traditional academic background. I'd worked in pharma and large CROs. I interacted with key opinion leaders, in the site relationship, but hadn't really gotten behind the scenes. I think my experience over the last decade in working closely with an academic team is, I think that there's a tremendous opportunity to innovate, and that when you can set aside, perhaps individual objectives and focus on a broader purpose, you can bring together great teams of motivated individuals with great minds. And when you pair that with private partnership and an opportunity to invest and provide resources, operational resources, I think it makes quite an attractive combination.
Okay. Bill, I wanted to come back to, you had a long list of potential opportunities that we should be thinking about advancing. And let's just think about Crohn's disease or its variants thereof. The post-op space, the pouchitis, some of that is Crohn's disease after the ileoanal pouch, and then fistula specifically. Could you just give us your thoughts about those indications in general, what needs to be done, and from both a investigative side and a regulatory side to really successfully get drugs approved for those indications?
Well, for some of you will remember the sort of forward-looking clinical trial that was done with infliximab, must have started a decade or so ago, in post-op Crohn's. And at that time, the negotiation with the regulators indicated that you had to have co-primary endpoints to show differences in both the clinical remission rate over 18 months or so and in the endoscopic recurrence rate. And at the end of that study, what happened was there was a robust difference in the rate of recurrent ulcers, much lower with long-term infliximab, but the differences in the clinical remission outcome were less and were not statistically significant. So the trial failed and the drug was not approved for that indication. I think everybody, including maybe the regulators, learned something from that. And I've been in some recent discussions with the regulators where there is more of an openness to discussing having the primary endpoint be endoscopy. However, what I heard from that meeting was that they would like a much better handle on the natural history of what the benefits that come with preventing endoscopic recurrence. So there's an opportunity and sort of an obligation for the field, and Robarts could play a role in this, to demonstrate that if you prevent ulcers after surgery, that really is a biomarker, if you will, for prevention of clinical recurrence and surgery and other things down the road. So there's definitely room for innovation in that outcome measure. In pouchitis, a trial was completed recently, and many of us played a role in that with central reading, looking at endoscopy and clinical factors in pouches. And the trial was not successful, but we learned something about the outcome measures as well. And I think our thinking is evolving about how to look at that. I think many of us feel that it may be that a PRO of stool frequency and histologic inflammation versus endoscopic findings might be the better sort of dual outcome measure for pouchitis. But of course, we got to go prove that. But I think the architecture of outcome measures for pouchitis is in its infancy. And the more that we figure that out, the more that it becomes an investable entity. So, and that's essentially what I was saying previously, is you've got to identify measurable outcome measures that are responsive to change and that regulatory authorities are convinced are clinically meaningful to bring these sub-indications forward. Mild to moderate Crohn's is another example that used to be just based on symptoms. Now you would need to find something objective. So does it need to be endoscopy? There's some data that histology actually correlates better with abdominal pain for ileal Crohn's disease than endoscopy. Well, that might be interesting. Could you have co-primary endpoints of abdominal pain and histology rather than endoscopy for mild to moderate Crohn's? And then for fistulas, the drug that's approved, infliximab, was based on this physical examination of the perianal fistula tracts. Now MRI is required. We showed the tagline of the paper that a number of people worked on to develop this MRI fistula index. We need to be test driving it in some fistula trials. I think we will see more fistula trials. There's still a learning curve probably going forward to understand the interaction between the MRI endpoint and clinical outcome measures, but it clearly remains an unmet need. The stem cell activities are going to be an important advance, but they're not really designed to treat active perianal Crohn's disease. They're designed to treat sinus tracts after you've controlled the inflammation. So studying fistulas in the context of active disease will be really important. It's a big opportunity. And then, we could say the same thing about structuring Crohn's disease, even mild to moderate ulcerative colitis. Historically, the Mayo endoscopy score of 1 was in that space. Well, I think the Mayo endoscopy score of 1 is a heterogeneous entity. Some of them will be biopsy positive for histology by the Robarts Histopathologic Index, and some would not be. So should we be thinking in mild to moderate ulcerative colitis of going down to Mayo 1, but only if you're histology positive? Well, that'd be sort of innovative, and it would give us a way of building out oral drugs that are post-mesalamine and pre-conventional biologic space. And so there's a lot of opportunities, I think, for building out these niches. And in the end, every one of these things is about the outcome measures.
Okay, great. So there's hope after all.
I think so.
Great. Well, I'd like to thank everyone for joining us today. I want to thank our guest speakers, Dr. Sanborn and Duncan Hall. Thank you very much for your time and for your presentation today, and our panel leader, Dr. Fagan. We would encourage those who are watching to please go on our new website, alimentive.com. Look for our new emails, which have changed over today. We're excited about the future that we have, and we thank everyone again for joining us today. Have a great day, everyone.